السلام عليكم الطلبة الأعزاء ورحمة الله وبركاته. محاضرة اليوم تتعلق باثنين من Medically Important General Bacteria which are the Proteus and the Pseudomonas. Starting with the Proteus species. Proteus are a member of normal intestinal microbiota and opportunistic pathogen. It is also a member of the Enterobacteriaceae family, and this genus is divided into four important species, which is Pseudomonas Proteus mirabilis, Proteus vulgaris, Proteus mixophaciens, and Proteus spineri. Proteus mirabilis and Proteus vulgaris are the medically important species as they are causing a wide range of infection for a human. So the morphology and the cultural characteristics of Proteus, there are some important features related to this bacteria. Starting from the active motility of this bacteria. Bacteria is very active motile, and we are growing on the agar plate. They are forming a swarming growth. Also, the bacteria is gram-negative as a member of Enterobacteriaceae. They are non-encapsulated. They are aerobes and facultative aerobes. As the bacteria is active motile, sometime non-motile variants can be isolated in some cases as they are losing their flagella. So, although the bacteria is very active motile, and this is the main feature for detecting them, but sometimes non-motile variants can be isolated. So, the bacteria also growing well on the ordinary media such as the nutrient agar. Another important feature that can help us to identify this bacteria is the fishy odor that they are produced when they are growing on agar. So this fishy or seminal odor is a kind of another feature that we can use it to recognize the Proteus species when they are growing on the agar plate, such as the nutrient agar and the blood agar. This is in a combination with the swarming growth, so it is make it very easy to identify and characterize the Proteus bacteria.
The swarming growth of the Proteus, as we can see from this picture, these are three colonies of the Proteus mirabilis on the agar plate. And how can we see that there is a kind of rings or waves spreading away from the center or the margins of each colony into the surface of the agar plate. This is called the swarming growth, and it is, as we mentioned, a ring or waves of transparent films composed of the bacterial growth spreading from the margin of the young colony on the surface of the plates. This is a characteristics feature of the two species of the Proteus mirabilis and the Proteus vulgaris, and it does not happen in the other species of this genus. This phenomenon actually comes from the vigorously active motility of this bacteria. And this is, as we mentioned, it is well known for such feature. They are very active motile bacteria, and this is maybe cause a problem in the lab when growing of the Proteus in a mixed culture with other bacteria. It can cover most of the agar plate and also the surface of the colonies of the other organisms. Therefore, in some cases, we need to inhibit the growth of swarming for specific requirements, such as detection of another bacteria in the mixed culture, as we can see in the next slide.
So the swarming growth can be inhibited by different ways, such as increasing the agar concentration of the media to around 6%, or also by adding chemicals additives to the media, such as phenyl ethyl alcohol. So these are two different ways for inhibiting the swarming growth. If you want to do that for different purposes, such as encouraging the growth of other bacteria on the same plate. The bacteria also do not form swarming on McConkey agar, and as the bacteria are non-lactose fermenters, so their colonies will be colorless. I mean, there is no change to any pink color, and they are smooth. At the same time, the non-motile variant of the swarming growth will not do any swarming growth, as the swarming phenomenon is a result of the active motility of the bacteria and separating them away from the starting colonies and making the waves, as we have seen in the previous slide. So the non-active motile not do not forming any swarming phenomenon. The biochemical reactions, so there are some of the different biochemical reactions that can be used to characterize and identify the Proteus species. Starting from the urease, it is positive for this enzyme. This is also a kind of important feature for the Proteus, as a production of this enzyme playing an important role in the pathogenicity of this bacteria. So it is positive for the urease, and also they are producing acid from glucose fermentation. They are non-lactose fermenter. For the indictase, they are indole positive by the Proteus vulgaris is positive for the indole, but the mirabilis is negative. Methyl red is positive for both the species. Volcostopus negative. Citrate is positive for mirabilis but negative for vulgaris. They are producing H2S, and they are reducing the nitrate to nitrite. So these are some of the biochemical tests that can be utilized to for testing and identifying the Proteus species. The antigenicity or the antigenic structure of Proteus. The Proteus as a member of Enterobacteriaceae. It also has the O antigen and the H antigen, but no K antigens. So starting from the O antigen, this antigen is also used to grouping or serogrouping the Proteus species depending on the variability in this surface structure. There are about 32 groups have been characterized for Proteus mirabilis and 22 groups for Proteus vulgaris, and the total is 54 groups for on the basis of their antigens. So it is similar in the other enteric bacteria such as E. coli, Klebsiella, and also Salmonella. The O antigen is used for serogrouping the Proteus species into different serogroups. The H antigens also has been used for classifying and grouping the bacteria. It also using for differentiate the different strains to different serotypes. So we have serogroups, serotypes, according to the variability in this antigen. And there are about 19 different H antigen in the mirabilis and Proteus vulgaris. While the K antigens, as the bacteria is lacking the capsule, so there is no K antigens and there is no utilization for classification of the bacteria depending on these antigens. So Proteus pathogenesis. This bacteria is opportunistic pathogen to human, as it is a member of normal human microbiota. They are one of the common causes of urinary tract and also septic infection, and the type of infection tend to be nosocomial. Mirabilis species are responsible for the majority of the cases of human infection, 90%, while the vulgaris is always or often considered as a common cause of nosocomial infection. The different type of infection that can be caused by this bacteria involved UTI, wound infection, pneumonia, infection of the air, respiratory tract infection, and septicemia. The UTI infection caused by Proteus tend to be more serious in comparison to the UTI infection caused by E. coli and other bacteria. And this is actually due to the urease enzyme that produced by the Proteus. As we mentioned, this enzyme play an important role in the virulence activity of this bacteria, as it lead to the hydrolyzing the the urea and the urine and generating ammonia and carbon dioxide. So the ammonia lead to change the pH of the urine to and make it alkaline pH, and this lead to precipitation of the calcium and magnesium salts from the urine and then producing of urinary stone. So this is one of the main serious complication caused by the UTI infection with Proteus. The also production of ammonia can lead to inactivating the complement system and then making a damage to the renal epithelium. It also may lead to hyperammoniamic encephalopathy and coma. These are another important complication can be resulted or generated from the infection of the with the Proteus. This bacteria can also colonize the bladder in patients with long-term indwelling urinary catheters. For laboratory diagnosis, starting from specimens collection, it depends on the type of infection caused by Proteus. The specimens can be urine, blood, pus, and also air discharge. Smear as a second step, gram staining. We can do gram staining and for testing the smear under the microscope, we will notice in the case of the Proteus infection, a gram-negative loss can be detected in the specimens. After the smear, we can do culturing. Uh, we can use different culture media, the same that have been used for other enteric bacteria, such as McConkey agar, and for in McConkey agar we can notice the colorless colonies Proteus as they are non-lactose fermenter. Also, we can use the nutrient agar and we can watching or monitoring the swarming growth from the Proteus, and also we can use EMB and blood agar. So, depending on their morphology and also culturing features, we can identify uh, the uh, the Proteus species in the clinical specimens, and also we can do another test, including biochemical test and agglutination test using a specific sera targeting the antigens for more specific characterization and identification of Proteus bacteria. Well, regarding the treatment, the different strains and the different species of Proteus are showing a strong variability in their susceptibility to the antibiotic therapy, but in most cases they are often sensitive to a wide range of antibiotics, including the penicillins, cephalosporins, aminoglycosides, and also imipenem. But the aminoglycosides and the cephalosporins tend to be the most active antibiotic agents against the Proteus species. The Proteus vulgaris in general tend to be more resistant to some antibiotics in comparison to other species, especially the ampicillin and the amoxicillin. Monas group, it is a large group of gram-negative aerobic non-sporing bacilli. The bacteria is also active motile using the polar flagella. Most of the bacteria belong to this group are saprophytes and found widely in soil, water, and other moist environments, and some of them are acting as pathogens for plants, insects, and animals. And also a few of them are responsible for many for many infections in human as opportunistic pathogen. The species has been divided into more than the genus, sorry, has been divided into more than 100 species. But in the latest study, a new taxonomy has been made for this group, and most of these species have been allocated to many other genera, including Burkholderia, Camamona, Stenotrophomonas, and Ralstonia. Well, among different species belong to the genus Pseudomonas, the Pseudomonas aeruginosa is the most medically important species that is most commonly associated with the human infection. This species is widely distributed in the nature and it's commonly present in moist environment of the hospitals. So it's responsible for a wide range of opportunistic infections in different sizes of a human with abnormal host defenses. As a typical organism, uh, Pseudomonas aeruginosa, as we mentioned before, motile using polar flagella, rod-shaped gram-negative bacilli. The aggregation of bacteria under the microscopic examination, the bacteria can be occur either as single bacterium in pairs and sometimes in short chains. But this is not an obvious feature for differentiation of this bacteria from other species or from other genera. So we have to do many other uh, identification tests in the lab for getting a specific identification or perfect identification for this bacteria. The culturing is another way for a better identification, including this bacteria can grow well on a wide range of culture media, including nutrient agar, maconchi agar, and bread agar, and many others. On the nutrient agar, the colonies can be obvious because it tend to be large, 2 to 3 millimeter in their diameter. They are also smooth, translucent, irregular, and they're round. Uh, the colonies also produce or emit sweet odor or grape-like odor. This is a kind of feature for this bacteria. It is obvious, so it's a kind of differential feature along with other feature, which is including the pigment production, and also some strains produce hemolysin. Then we can differentiate them from the non-hemolysin producing bacteria. The Pseudomonas aeruginosa is also produced a different kind of pigment when they are growing on the agar media. These pigments can be utilized as a characteristic feature or identification features for these organisms. The four pigments involve the biocyanine, which is the non-fluorescent bluish pigment diffused into the agar. This pigment is produced by the Pseudomonas aeruginosa and other species of Pseudomonas. And the bioviridine, which is a fluorescent pigment, gives a green color to the agar. And this is produced by some strains of Pseudomonas aeruginosa. There is also biorubin, which is a dark red pigment, and biomelanin, which is a black pigment. So these four pigments produced by Pseudomonas can be utilized as characteristic features and identification features for Pseudomonas aeruginosa. We have also another important feature is that clinical isolates of this bacteria are isolated from patients with cystic fibrosis. They are often form mucoid colonies due to the overexpression of exopolysaccharide, which is composed of alginate polymers, and it is known as a glycocalyx capsule. This glycocalyx capsule or alternate polymer play an important role in the pathogenesis of this bacteria, and it's possibly act as a matrix for the biofilm formation by the organisms, and also it is important for evasion of host defense, especially during the chronic pulmonary disease of patient with cystic fibrosis. So these are two pictures showing the different pigments produced by the Pseudomonas aeruginosa and the cetrimide agar agar. This is a kind of culture media used for Pseudomonas aeruginosa culturing. So in the right one, we have the fluorescence pigment produced by this bacteria under the UV light, while the one in the left is the showing the biocyanine pigment on the certamide media, which tend to be blue to the green color on the media. Another important characteristics for these organisms is their growth features. One of them is the ability of this bacteria to grow and multiply in a wide range of temperature ranging from 6 to 42 degrees. But the optimal temperature for their growth is 37, and the ability of this bacteria to survive and grow and as the 42 degree can be used as a differential feature to differentiate the Pseudomonas aeruginosa from the other Pseudomonas species within the fluorescent group. The bacteria is positive for the oxidase test, and they do not ferment the carbohydrate. So the identification of this bacteria is mainly depend on some feature as mentioned, including the colonial morphology, the oxidase positivity, the pigment production, and the ability of these bacteria to grow at 42 degrees. The antigenic structures and the virulence factors. So there are some surface antigens that can be used for the pathogenicity, or can be act as virulence factors for this bacteria, and can also be used for typing purposes of this bacteria. So starting from villi, which is mediates the attachment of this bacteria to the host cells. Also the lipopolysaccharide, which is a variable immunotype. This is part of the cell wall, and the art is a variable. It's a kind in similar way in the O antigen in other enteric bacteria. So we can use this antigen for typing the different strains of Pseudomonas aeruginosa. So we have variable immunotype and the variable structures of this polysaccharide. This also responsible for the endotoxic activity of the bacteria, as we mentioned, as it is a part of the cell wall. The Pseudomonas can also be typed. This is for the typing purposes. The bacteria are differ in their susceptibility to the biocene, which is a kind of bacteriocene. So the different strains can show different ability to resist to the biocene or susceptibility to this bacteriocene, and they can be grouped depending on this feature. The bacteria possess or produce extracellular enzymes, uh, especially the by the clinical isolates of Pseudomonas aeruginosa. These enzymes can play a role in the pathogenicity of this bacteria. Some of them including the elastases, proteases, and also hemolysins. We have exotoxin A. This is a toxin produced by this bacteria. This toxin leading to cause a necrosis of the tissue, and it is a lethal toxin for animals when they inject it in a purified form. The toxin is immunogenic. I mean, the human body can generate antibodies against this toxin and then neutralize it. And also, the mechanism of action of this toxin is similar to the mechanisms of diphtheria toxin mentioned in the previous lectures, as it leads to the inhibition of protein synthesis in human cells. The pathogenesis of the bacterium Pseudomonas aeruginosa. This bacteria is an opportunistic pathogen, and they are causing infection only in the case when they enter the body sites that are lacking the natural immune defenses. In many cases, including during the disruption of skin and mucous membrane by direct tissue damage, such as in born wounds. Also, in using of during using of intravenous or urinary catheters, and also during neutropenia, such as in cancer chemotherapy. The bacteria as a first step in during the infection, when the bacteria enter such sites of the body, they're using their virulence factors, starting from the adhesion factors, and in the case of Pseudomonas aeruginosa, they're using the villi as adhesion factor to attach to the mucous membrane or the skin, and then for colonize them. So as we mentioned before in the previous lectures, the first step of making infection is the colonization. So the bacteria using such structure for such purposes. So they colonize these sites and then invades locally, and then subsequently causes systemic infection. The enzymes and the toxins produced by this bacteria promote the local invasiveness and then the dissemination of bacteria into the different body sites. Endotoxic activity of the lipopolysaccharide leading to develop fever, shock, oliguria, leukocytosis, and leukopenia. So the bacteria can cause a wide range of opportunistic infection, starting from wounds and bones infections, meningitis, and the route of entry of this bacteria to the human body is during the lumbar puncture and also during neurosurgical procedures. The urinary tract infection, and this when the bacteria introduced by the catheters and instruments using for the patient, and also necrotizing pneumonia, mild otitis externa in the swimmers, and invasive malignant otitis externa in patient with diabetes, and also eye or corneal infection occurs after injury or surgical procedures or after using contact lenses and may lead to a rapid destruction or to die. Pseudomonas aeruginosa can also cause a systemic infection when they invade the bloodstreams and causing sepsis. And this happens in patients with leukemia or lymphoma who have received antineoplastic drug or radiation therapy, or in people who with severe burns. And the nature of this infection tend to be a lethal sepsis. Another infection is the eczema gangrenosum, which is a hemorrhagic necrosis of skin surrounded by erythema and often do not contain pus. This infection occur either alone without any previous infection or due to the sepsis infection caused by Pseudomonas aeruginosa, and this happen particularly in people who are suffering from leukemia and other malignant diseases. Pseudomonas aeruginosa then can be detected in such infection by making a gram stain for the specimens taken from the lesions or by culturing the specimens on other culture media and doing some further tests. The symptoms of infection with Pseudomonas aeruginosa are varied greatly depending on the infected site and the infected organs. The diagnostic laboratory test using for identifying and characterizing the Pseudomonas aeruginosa in the clinical samples. So starting from the first step, which is the specimens collection. The specimens are vary greatly depending on the different body sites and depending on the type of infection. So it can be pus, urine, blood, spinal fluid, and sputum. So depending on the type of infection and the body site infected with this bacteria. From these specimens, we can do gram staining and the microscopic examination of the smear. And then we need to look for gram-negative rods, as this bacteria is a gram-negative rods. But this step is not a key step in the identification because the morphological features of the Pseudomonas aeruginosa under the microscope is not differ greatly from the enteric or other gram-negative rods. So we need to do further tests, including culturing the specimens on several culture media, starting from the media that used for the enteric bacteria, which were such as the McConkey agar, as the Pseudomonas can also grow on this media, but it tend to be slower than the enteric bacteria. But this media can be used for such purposes, and it can give us some indicators, such as when the growing on the McConkey agar, the bacteria is non-lactose fermenter, and then will produce the colorless colonies. Using a nutrient agar is also useful for such purposes, I mean for identifying, because the bacteria will grow on this agar and then produce the blue-green pigment, which is a specific feature for these organisms, and also produce or forming the fruity odor. The treatment of the infection with Pseudomonas aeruginosa, there are several aspects that should be taken into account when using the antibiotic therapy against this organism. Firstly, the bacteria is showing a considerable natural resistance to a wide range of antibiotics, therefore using a single antibiotic against it, leading to a rapid development of a resistance strains by this organism. So, using a combination of extended spectrum penicillin and also an aminoglycoside is recommended for such purposes. There are many other clinically effective antibiotics can be used, including the carbapenems, such as imipenem or meropenem, and the fluoroquinolones, including ciprofloxacin, as well as cephalosporins. The susceptibility patterns of the different strains of Pseudomonas aeruginosa vary geographically. So, doing the susceptibility test in the lab should be done as an important step to decide which antimicrobial therapy is suitable to use against the infection with this bacteria. The epidemiology and control of Pseudomonas aeruginosa. As we mentioned before, this organisms is widely distributed in the nature, and there are two main features enable the bacteria for such wide distribution. The first one is the ability to resist to many antibiotic therapy, and also their ability to grow at a wide range of temperature. Therefore, the bacteria are able to colonize a variety of environments, including the moist equipments in hospital, wards, bathrooms, and kitchens. The bacteria also can transiently colonize the respiratory and the gastrointestinal tracts of the hospitalized patients, and this also help in spreading this bacteria within the hospital area. So measures that can be taken to control the spread or the infection with this organisms is the same as any measures that can be taken to control any other nosocomial pathogens. So more attention should be paid for sterilizing the hospital equipments, apparatus, tools, and dressing, and also avoiding the excessive and inappropriate use of broad-spectrum antibiotics. For epidemiological purposes, the strains of Pseudomonas aeruginosa can be typed depending on molecular typing techniques. This is the end of our lecture, and thank you attention. Thank you for your attention.